Welcome back to my channel Knowledge Chronicle.
In today's video, we're diving deep into a topic that affects the very food we eat, the crops we grow, and the future of farming, biotechnological applications in agriculture. Whether you're a student, an aspiring scientist, or just someone who's curious about how science is revolutionizing our farms, this is for you. What is agricultural biotechnology? So what exactly is biotechnology in agriculture? Biotechnology involves using living organisms, like microbes, plants, fungi, or even animals, on an industrial scale to produce useful products. These could be biopharmaceuticals, vaccines, food additives, or genetically improved crops. The three main research pillars in biotechnology are: first, improved catalysts, usually microbes or enzymes that speed up biological reactions. Second, engineering optimal conditions, so the biological catalyst can work most effectively. And third, downstream processing techniques used to purify and recover useful compounds like proteins or organic molecules. But how does this relate to agriculture? Well, biotechnology is helping us solve real agricultural problems, from crop diseases to soil fertility, and from food shortages to malnutrition. Let's begin by looking at how we've traditionally tried to increase food production and where biotechnology fits in. The three traditional approaches to increase food production. Historically, we've had three broad options. First, agrochemical-based farming, heavy use of fertilizers and pesticides. Second, organic agriculture, more natural but slower in impact. Genetically engineered crops are, well, all about using science to create better seeds and plants. The Green Revolution did help in tripling food production, but it mostly relied on agrochemicals and high-yielding crop varieties. However, these chemicals are expensive and often harmful to the environment. Also, conventional breeding is slow, often taking years to develop a new variety. So, what next? Tissue culture and micropropagation, cloning nature in test tubes. This is where tissue culture changed the game. In the 1950s, scientists discovered something fascinating: totipotency. Totipotency is the ability of a single plant cell called an explant to regenerate into a complete whole plant. Yes, you heard it right. A single cell can become a full tree. Here's how it works. An explant, which is a part of a plant like a leaf or root tip, is placed in a sterile test tube. It's grown in a nutrient medium that contains sucrose as a carbon source, inorganic salts, vitamins and amino acids, and plant hormones like auxins and cytokinins. So, this technique is called micropropagation, and it actually lets us grow thousands of genetically identical plants, which are known as somaclones, in a really short time. Real-life examples. Well, banana, apple, tomato, grapes. All of these have been commercially multiplied using this method. It's also super helpful in producing virus-free plants. Even if a plant is infected, the meristem tissue, which includes the apical and axillary buds, often stays virus-free. By culturing just the meristem, healthy plants can actually be regenerated. This approach has saved entire plantations of sugarcane, potato, and banana from viral diseases. But, biotechnology doesn't stop there. What if you wanted to combine the traits of two completely different plants? That's where somatic hybridization comes in. So here's how it works. Plant cells are taken and their cell walls are digested, which leaves just the protoplasts. Basically, these are naked cells with only the membrane. These protoplasts are then fused together using chemicals or electric pulses. The hybrid protoplast is then grown under lab conditions into a full plant. These are called somatic hybrids. For example, scientists actually combined a tomato and a potato to create the pomato, a plant that grows tomatoes above ground and potatoes below. It sounds amazing, but commercially it lacked some important characteristics. Still, it really showed what's possible using somatic hybridization. Genetically modified organisms, or GMOs, are basically about redesigning nature. Now let's talk about the most powerful tool in agricultural biotechnology: GMOs, or genetically modified organisms. These are plants or animals whose genes have been altered using genetic engineering. The result? Well, you get crops that can withstand stress, resist disease, and even produce nutrients. The benefits of GM crops include, first, resistance to abiotic stresses like heat, cold, drought, and salinity. Second, pest resistance, so farmers need fewer chemical pesticides. Third, lower post-harvest losses, which means better shelf life and less spoilage. Fourth, efficient use of nutrients, which helps reduce soil damage. Fifth, improved nutritional value. For example, golden rice, which is rich in vitamin A, and there's more. GM crops are now being used to produce industrial materials, bioplastics, and even medicines. Beet crops offer built-in pesticide protection. One of the most widely used GM technologies is Bt crops, especially Bt cotton. Bt stands for Bacillus thuringiensis, a soil bacterium that naturally produces protein crystals toxic to insects. Here's how it works. The Bt toxin protein is produced as a protoxin, which is an inactive form in the bacterium. When an insect like a bollworm eats the Bt cotton, the alkaline pH in its gut actually activates the toxin. The active toxin binds to the insect's gut lining, creates pores and causes cell death, killing the insect. Bt crops include Bt cotton, Bt corn, Bt brinjal, rice, tomato, and soybean. Different Bt toxins target different insects. For example, Cry1 and Cry2 AB control cotton bollworms, while Cry1 AB targets the corn borer. This drastically reduces the need for chemical insecticides, protecting both the crop and the environment. Now, let's explore one of the most intelligent biotech strategies, RNA interference, or RNAi. This is actually a natural gene silencing mechanism found in all higher organisms. It's kind of like the cell's internal defense system. It shuts down harmful or unwanted genes. So, here's how it works. When double-stranded RNA or dsRNA enters the cell, it actually triggers a process called RNA interference or RNAi. The dsRNA binds to a complementary messenger RNA or mRNA and blocks it from being translated. This, in turn, stops the production of specific proteins, effectively silencing the gene. In agriculture, this has been used, for example, to protect tobacco plants from a root-infecting nematode called Meloidogene incognita. Scientists introduced nematode-specific genes into the tobacco plant using agrobacterium vectors. These genes made both sense and antisense RNA, which then combined to form dsRNA inside the plant. This dsRNA initiated RNAi and silenced the nematode's essential genes. As a result, the parasite couldn't survive, and the plant stayed healthy. No chemicals, no pesticides, just smart genetics. The future of food is honestly all about biotech. From tissue culture to genetic engineering, from Bt cotton to RNAi, biotechnology is totally revolutionizing how we farm, how we feed, and honestly, how we survive. It's not just about growing more, it's about growing smarter, safer, and more sustainably. As we face climate change, population growth, and soil degradation, agricultural biotechnology gives us the tools to meet these challenges head-on. If this video helped you understand the power and potential of biotechnology in agriculture, then your one like, share, and subscribe will help us create more valuable content for you. So, stay tuned, stay informed. This is Knowledge Chronicle, and I'll see you in the next one.